5 minutes later.
Poor Shafi, I bet from the incident this is the best reason why we should study about this topic. Unlike Shafi, have you ever noticed a cottony growth on leftover bread? Actually, it is not only happens on the bread, it occurs on many other food, especially when they are moisturized. Have you ever wondered what makes them there, or what actually they are made of? Well, it is actually a set of live cells. By now, you must have guessed what is the name of these organisms. Yes, it is fungus or fungi for the plural. So fungi can be broken down into two parts, which are microscopic and macroscopic. So microscopic when it can be observed under a microscope, and macroscopic when it can be observed with naked eyes. Later, Shafi will explain to you about the similarity of both of them. So today, we are glad to share with you about the evolution and characteristic of fungi. Fungi were the first eukaryotes that invaded land. They are ancient organisms. Fungi are whole separate kind of living thing. They are neither animals or plants. Fungi are classified as living organisms because they carry out the vital function of interaction, reproduction, and nutrition. Fungi come from different kingdoms with millions of species coming in thousands shapes and sizes. Some might look like mushrooms, but others are weirder. There is a black witch's butter, which is a type of fungus called a jelly fungus. Then there is a brain mushroom, which resembles a lot like a brain, obviously. There are even mushrooms that glow in the dark. Fungi might seem like plants from the outside, as many of them grow from the ground. But scientists have made some pretty cool discoveries about fungi that makes them different from plants. Fungi are in the supergroup of Opisthoconnta. Fungi includes over 140,000 species of mostly multicellular eukaryotes that share a common mode of nutrition, which is saprotrophic, which I will explain in detail sooner. Just don't forget that eukaryotes mean that it has nucleus and organelles. Plants are autotroph, in which they create their own food by the process of photosynthesis. Conversely, fungi are like animals. They are heterotroph, which cannot produce its own food. Instead, they take nutrition from other sources of organic carbon, mainly plant or animal matter. However, animals are heterotrophs that ingest food, while fungi are saprotrophic, which organisms that absorb food. The hyphae, or the structure of fungi, will secrete digestive enzymes into the immediate environment. It will then break down dead and decay the organic matter outside of their body. The resulting nutrient molecules are then absorbed by the fungal cells and utilized by the entire fungal body called mycelium. Mushrooms, for example, eat dead plants, which is why they often grow on trees or the forest.
Where do we find fungi in our daily lives? Some fungi are edible and very tasty, like mushrooms and truffles. But be careful, some also might be toxic. There are also other foods like some types of cheese that have fungi in them, for example, Roquefort cheese. Some fungi are harmful to our health that may cause skin or lung disease. Fungi also produce antibiotic substances used as medicines to fight bacterial infections. One of the most popular and widely used antibiotics is penicillin. So, Alia has briefly explained to us about the characteristics of fungi. Now, we are moving on to the most important part of the topic, which is the evolution of fungi. Actually, the classification of fungal phyla is still under progress and yet imperfect. The scientists are still carrying out research and doing experiments about the fungal phyla. Do you still remember about the phylogenetic tree of 6 eukaryotic supergroups that we have learned in previous lecture? Before we get into this topic, I will recap a little bit of that. The phylogeny tree of eukaryotic supergroups consists of 6 supergroups, and they are Excavata, Chromaviolata, Rhizaria, Archaeplastida, Amebozoa, and Ophistoconta. Fungi are the members of supergroup Ophistoconta, which also includes the animal-like quanoflagellates and animal. Let us take a close-up of the fungi branch of the eukaryotic evolutionary tree with 6 recognized phyla and a 7th branch representing the arcetal lineage of the Zygomycota. The 6 fungal phylogeny consist of Plastocladiomycota, Zygomycota, Neocalimastigomycota, Crytridomycota, Glomeromycota, Basidiomycota, and lastly, Ascomycota. This evolutionary relationship represents a current hypothesis of fungal evolution. Remember when I mentioned that the classification of fungal phyla is still under progress and yet imperfect earlier? Well, this statement has been proven true, as recently there are 2 major changes in the hypothesis. Firstly, recent sampling has detected the existence of new fungus group. Previously, Neocalimastigomycota and Blastocladiomycota is recognized as a distinct phyla, so it has been categorized as its own group. Secondly, it has been discovered that there is a previously identified group that is not even monophyletic. Does everyone remember what does it mean by monophyletic? Monophyletic group of species shares a single common ancestor and also includes all of the descendants of that common ancestor. In simpler words, we can say that a monophyletic group comes from the same ancestor. In this case, phylum called Zygomycota is no longer recognized in current classification because of increasing evidence that it was not monophyletic. Instead, its member called Zygospore fungi are paraphyletic. What is paraphyletic? Paraphyletic is a group of organisms that descended from a common evolutionary ancestor group but not including all descendant groups. However, in this chapter, we are only going to link 2 major lineages, which are Ascomycota and Basidiomycota. Fungi are not animals or plants. They might seem like the plants from the physical appearance, as most of them grows on the ground too, like plants. Plants, animals, and fungus all have common ancestors in protists, although molecular evidence suggests that animals and fungi shared common ancestor more recently than animals and plants. As a result, mammals and fungi are more closely related to each other compared to plants. Scientists have discovered some facts about fungi that make them different from the plants. For example, one thing that makes the plants special is they make their own food out of sunlight. That is how plants do eat, but fungi can't do that. Instead, they have to get their own food from other living things, just like animals. That is also why you can see mushrooms growing in the dark places, as they do not need the sunlight, but plants will not grow in the dark. Animals and fungi are both likely the same because both are heterotrophic, which means that their source of feeding is by obtaining organic molecules from other organisms. Fungi is a saprophytic organism that feeds on non-living organic matter by secreting digestive enzymes and absorbing the product. An aquatic flagellated single-celled protist was most likely the common ancestor of animals and fungus. After animals and fungus were divided into 2 distinct lineages, multicellular creatures developed.
Fungi do not fossilize well. Due to that, it is difficult to estimate from the fossil record when it is first evolved. The first known fossil fungi was 460 million years ago, but fungi was evolved a lot earlier. While animals were still swimming in the seas during the Silurian, plants were beginning to live on the land. They brought fungi with them. Mycorrhizae are evident in plant fossils, which are also dated 460 million years ago. Perhaps fungi were instrumental in the colonization of land by plant. Much of the fungal diversity has its origin in adaptive radiation when organisms began to colonize land.
Most fungi are multicellular, while the few rest are unicellular. This is a mushroom, a model mushroom. It is an example of a multicellular fungi. It's got a cap which is at the top, hence the name cap. The skirt-like structure around the stem, which is called annulus. Now, let's get to the interesting part. This is the same mushroom, but it is cut in half so we can see the insides of a fungi. I'll be using this model to explain the structure of fungi. The body of most fungi is made out of mycelium, which is a network of fungal filaments called hyphae, or for a single strand, we call it hypha. Hyphae is like a bundle of delicate strings because this kind of structure will provide quite a large total surface area to volume ratio to increase the absorption capability. Now with the model out of the way, I will show you how hyphae grow. Imagine this piece of plasticine is hypha. Hypha grow from the tips like this. And in some fungi, a wall of tissue called septa will form behind the growing tip like this. These fungi with septa in their hyphae are called septate, while fungi without septa in their hyphae are called aseptate. These walls have pores that allow cytoplasm and sometimes organelles to pass freely from one partition to another. As a side note, although fungus cells and plant cells appear similar, they are actually quite different. Fungus cells lack chloroplast. Their cell wall are made of chitin and not cellular. They store energy in the form of glycogen instead of starch, just like animals. Now, like Alia said, fungi can be classified in two different categories, which are the macroscopic fungi, the bigger ones, and the microscopic fungi, the smaller ones. And by small, I mean very small, that you have to watch using the microscope, like the mold on your bread. But do take note, even though they look very different, they are still fungi, which means that they share the same basic structure of a fungi, mainly their body made out of mycelium, which is formed from network of hyphae, just like this.
Next, we will look at the reproduction of fungi. So basically, reproduction is the biological process by which new generation variation organisms are produced from their parent. The known methods of reproduction are broadly grouped into two main types, which is sexual and asexual. The majority of fungi can reproduce both sexual and asexual reproduction. This allows them to adjust by conditions in the environment. Okay, let's go to the first reproduction in fungi, which is asexual reproduction. Most fungi are using this type of propagation to generate a new genetic variation among them. The parent undergo mitosis, produce a huge number of spores at a time to produce new individuals of fungi. In fragmentation, the mycelium breaks into segments of hyphae. Each segment, by further division, will develop into new mycelium. Or by budding. For example, in yeast, it involves the formation of a small bulge on the side of the parent cell, and it gets pinched off when it grows to the full size. After that, another propagation in fungi is sexual reproduction. In sexual reproduction, fungi can increase their genetic variation when conditions in the environment are changing, and it may help them to adapt to the environment. Sexual reproduction also occurs in virtually all fungi. During mating, two different haploid hyphae, which is the parent cell, fuse, form a diploid spore N plus N, called dikaryotic hyphae or zygospore. The zygospore is genetically different from the parents. After the zygospore germinates, it can undergo meiosis, forming spore haploid cell that develop into new hyphae. This is the spore look like under the microscope. Just some facts that you must know. Some parent cell fuse immediately and some fuse for days, months or years. Lastly, in order to prevent the new generation of fungi compete with their parent in space or other resources, the spores that produce from sexual or asexual reproduction will be dispersed by moving water, wind or other organisms. Then they can develop into new hyphae, which is new fungi.